Hello and welcome back to microbiology. Today we're going to talk about chapter 6, which is really our introduction to viruses. Viruses are very important to us and a topic that we'll definitely pay attention to because viruses can cause infection. But one thing people don't understand is that viruses can infect really every type of cell in existence. Things like bacteria, algae, fungi, protozoa, plants, even animals. It's important to understand that typically though, most viruses will only infect one cell type, meaning that the bacterial virus will only attack bacteria, and the virus that attacks algae should only attack algae and not plants. So really there are still lots of questions out there about what viruses are. Are they organisms? Are they alive? How did they affect evolution and the change in characteristics of life? There have been, again, lots and lots of discussions about these topics. But overall, we'll talk about each of these points as we go through our discussions together. There are several unique properties of viruses that we're going to have to discuss before we can get too far in, including how viruses are infectious particles instead of being organisms. And organisms are considered to be alive, and since viruses aren't alive, they can't be organisms. Naturally, again, if you're not alive, you can't be living or dead. So instead we refer to them as being active and inactive. And active viruses can cause infection. One thing to note that is an absolute is viruses are obligate intracellular parasites. They must have a host to reproduce them. So overall, you can look through this list of properties. Again, viruses aren't alive, they're not cells. They have to have a host. They don't and basically meet all the characteristics of life. They're really just inactive until they find their host. Again, they do nothing at all outside of a host cell. They're ubiquitous in nature, causing, again, infections in a wide variety of different organisms. They have DNA or RNA, but never both. They carry, again, surface features that recognize their host cells. And so the idea here is, again, there are lots of unique features that you guys can kind of go over and look through whenever you guys try to start learning about these viruses. So one of the things that we should really point out here is that the viruses tend to be very small. Okay, so we did this earlier with kind of a comparison between eukaryotic things and bacteria and viruses, but notice now we're just looking at specifically our yeast cells, which are small, bacteria like E. coli and strep and rickettsia, and now we start looking at things like viruses here. And so you see viruses range from being roughly the size of bacteria, but can be as small as something like hemoglobin, which is a protein really found on the surface of red blood cells. And so the idea here is these viruses can be very different in terms of size, but they're all very small is the bigger idea. So in terms of what viruses look like, they don't really look like anything else we know about. And it's because they don't have the parts required for life. They don't have the, again, structure that living things must have. Because they're not alive, they don't follow the same rules. And so really what viruses have is just typically genetic material and protein. Speaking of protein, the capsid is actually going to be made of proteins. And its job is to actually take and protect the genetic material we're going to call nucleic acids. Some viruses do have a structure called an envelope, and this envelope is a membrane that has been stolen from the previous host, and so it's going to actually cover itself whenever it leaves the host with the host cell membrane. And so it's important that everything is already put together as that virus goes to leave the host they were made in, so that they can steal the membrane as they leave. Whenever a virus only consists of a nucleocapsid, we refer to that thing as being a naked virus. Another important feature that we should discuss is spikes. Spikes is going to be found on really most viruses, and what spikes are used for is recognition and attachment. So again, it allows them to dock with their host cell, but also again recognize their host cell. So I want to point out a bit of a misconception And it's the word virus is often misused. Technically, a virus only exists inside of a host cell. Whenever it's outside of a host cell, it's really called a virion. But I won't hold you accountable for that because, again, really nobody follows that. So you can see here two different viruses, the naked virus and the envelope virus. Again, the naked virus we call naked because, again, it doesn't have anything more than a nucleocapsid. And you can see the capsid itself is actually going to be covering the genetic material inside and protecting it. This is an envelope capsid because, again, it has that membrane structure that surrounds the capsid, which is on the inside, which, again, the capsid covers and protects the genetic material. So what are capsids made of? Well, they're made of subunits called capsomeres. So these protein subunits are identical and actually form inside the host cell, but then spontaneously self-assemble to actually start manufacturing viruses as the viruses are about to leave. There are really two major types of capsids we're going to discuss: helical capsids and ixahedral. The helical capsids, again, form a helix around the nucleic acids, while the ixahedral makes a large 20-sided figure with, again, evenly spaced corners. So you can see here the helical nucleocapsid. The capsid is this tube here that surrounds the genetic material or nucleic acids. And so you can see it makes a helical structure as it wraps around the genetic material. Whenever it comes to nucleocapsids, you can have naked versions that are just on their own, or you can have envelope versions that are surrounded by, again, an envelope. The ixahedral, again, being a three-dimensional structure, is important to note that in that space inside, like seen here, we house the genetic material to protect it. But just like before, we can have naked ixahedrals or enveloped ixahedrals here. Some viruses actually have complex capsids, and these complex capsids, like this one of the bacteriophage, look incredibly scary, and so people often think that they can be infected by something that looks like this, but the reality is you can't. This is a bacteriophage, so it really only infects bacteria. Specifically, this is what we call a T-even phage that typically only attacks just E. coli. So the viral envelope is something we're going to spend a tiny amount of time. We already kind of discussed this with this idea that the envelope is the previous host cell membrane, but it's important to note that, as we said before, the membrane has to be completely prepared to become what is the viral envelope. So it's important to understand that once viruses leave their host, they do nothing until they find a new host. And so if we need spikes in the viral envelope, then the spikes have to be put in while the virus is still being assembled in their previous host. That way, whenever the virus goes to leave that host, it can pull the host cell membrane with it, creating now an envelope for the virus. So one of the things that is always going to have to be there for a virus is genetic material. That's really the important information that allows the virus to do what a virus does. And really, that genome can be either DNA or RNA, but never both. So with that in mind, it's pretty open beyond that one rule that you're either DNA or RNA. There are lots of different types of viruses in terms of their genetic structure. Some have RNA that we call positive sense, which means that is basically like messenger RNA and ready to be translated into proteins, typically at ribosomes. Negative sense RNA, on the other hand, is something that needs to be converted to positive sense before it can actually be utilized. So it's important to understand that negative sense, once it gets in the host, must be changed over to the equivalent of what would be like messenger RNA, and then that messenger can be turned into proteins later. So as I was kind of discussing before, this idea that you must have DNA or RNA, but other than that, it's pretty open. So notice we have some situations where viruses have double-stranded DNA, some that have single-stranded DNA, some again that are positive or negative sense RNA strands. We even see double-stranded RNA in some cases, and then even some single-stranded reverse transcriptase cases, like specifically HIV. We'll discuss this one a little more a little later. Viruses have additional substances inside of them, including things like enzymes for hijacking the host cell. Things like polymerases can help copy DNA and RNA, or an enzyme like reverse transcriptase can actually synthesize DNA from RNA. This is really important for something like HIV. HIV can actually take its RNA genome and turn it into DNA. They can then insert that DNA into the host genome, and it can lay dormant for a particularly long time. So classification of viruses come in a variety of forms. Informally, we'd like to classify them based off of what they infect, things like animals or plants or even bacteria. We also like to classify them informally, whether or not they're DNA or RNA viruses, what type of capsid they have, helical or ixahedral, even things like structures or chemical compositions. But one important thing to understand is for the classification of viruses, we have a particular naming pattern where we use viridae in reference to the viral family, and then we'll often use virus as being part of the genus name. And it's not uncommon to see things like letters and numbers being associated with the species level. So you can see this example here. Notice the family level, we have things like herpes viridae. Notice the genus is called simplex virus, again virus indicating it's a genus, and you can even see here that we're using at the species level a number to indicate which type of human herpes virus we're talking about. So here are some additional ones. Again, we're not going to stress a whole lot about the different types of infections right now. We'll spend a whole lot of time in that late stages of the class discussing infection. So again, for now, just understand that kind of naming dichotomy where the viridae is associated with the family, virus is associated with the genus, and then letters and numbers are often associated with the species. Again, more potentially dangerous human viruses, and still more. Did I mention we have more? Alright, now on to stuff again I do want you to definitely know for this particular test. There are some particular steps that viruses go through in reproducing themselves in animal cells, and those include really these 6 steps here: adsorption, penetration, uncoating, synthesis, assembly, and then finally release. An easy way to remember this is APUSAR. A for adsorption, P for penetration, U for uncoating, S for synthesis, A for assembly, and R for release. Here we can see those steps in action. Initially, the viral spikes need to attach to the host cell, allowing for adsorption or attachment. Now we need penetration to occur, where the virus is brought into the cell. Once in the cell, we start to unpackage or uncoat the genetic material. The genetic material can then be utilized to make new parts in a process known as synthesis. Once we have all the parts, we can assemble new virions or viruses. And then finally, the viruses release. Here we can see the same action, but again this time for a DNA virus, where we need to go to the nucleus to help produce the parts. So now we're going to go through some of the steps again in greater detail. One of the more important things to point out about the first step here is it is attachment, but we can use a fancy word adsorb to mean attachment. But do not say absorption, because that means to soak in, which is not right. So again, it's adsorption or attachment. If you don't feel comfortable with trying to remember adsorption, just call it attachment. We'll be fine. One of the more important things with adsorption is we need spikes to help recognize and attach to the host. But those spikes really help determine what is the host cell. So for instance, there are many examples of things with restricted ranges like hepatitis B. Hepatitis B has spikes that only recognize liver cells of humans, and as a result should only infect those. There are some that are a little less detailed. Again, poliovirus infects the intestinal and nerve cells of even primates like us. But there are even broad host ranges like rabies that can infect really nervous tissue of all mammals. But do notice, again, that's still a pretty small category. So overall, viruses are very specific in the hosts that they infect. So here's that penetration and uncoating. Again, penetration and uncoating are essential for basically gaining access to the host. And so once we penetrate or get into the host, we need to uncoat the genetic material, because really the genetic material is what we need to utilize to produce new viruses. Speaking of production, synthesis is all about producing the parts that we need, including more DNA or RNA and all the components like capsids and even spikes if necessary. So we can see again, this is an illustration of that synthesis where you can see all these different proteins laid down by the virus that can again help assemble into new viruses later. Release is really the next step we need to talk about. Notice this is an example that's illustrating the basic movement that allows for the production of an envelope. So as the virus is getting ready to leave, we prepare the membrane of the host cell by adding things like spikes. And as the virus goes to leave, it pushes through and steals or robs the host membrane to now produce the envelope that the virus needs. As you can imagine, all this activity inside of the cell actually produces damage, and we call that damage the cytopathic effects. That damage to the cell is typically permanent and can be seen in a variety of forms, including inclusion bodies and sentia. Here we can see again those effects, things like sentium, that's going to be again a mass of giant cells, and then inclusion bodies over here where we have the damage that's being present where the cell is really taking over and reproducing. This table is really intended to show you the different types of damage that can develop from a viral infection. There are things like inclusion bodies or even multinucleated cells that can form. The idea, though, is typically damage done to a cell is usually permanent damage, and that cell either lives or dies, but if again it lives, it is stuck with those ramifications. So let's talk about persistent infections for a little bit. Persistent infections are basically going to be a situation where a cell becomes essentially a carrier or a harborer of a virus. So we have two different scenarios here, the provirus and chronic latent. With the provirus, what's happening is the viral genome, like the DNA, is actually going to be inserted into the host DNA. And this is a survival strategy for the viruses in that they can be infecting a cell without actually showing the infection to the immune system. And as a result, we do have cells that are infected, but we don't see any active virions in the environment, and so the immune system doesn't recognize the infection. And so this is a pretty brilliant strategy, honestly, but the reality is we don't fully know the mechanisms behind it yet, and it's something that we are still looking into. Chronic latent is this idea that we can have these periods of activation after the inactivity, and so we have some viruses that can basically disappear from in terms of signs and symptoms, but then come back at a later point when given an opportunity to. So here's an example of an oncogenic virus. Onco is in reference to cancer, and so the idea here is these are potentially cancerous viruses, and we do think that the amount of cancers that are actually caused by viral infection specifically can be upwards of 13 to really 20% if we're being honest. And the idea here is that whenever a virus can actually take and incorporate its DNA into the host DNA, it can create a situation where it now is more likely to promote cancer. There are some situations where it can directly cause cancer, and as you can imagine, those things are very, very, very bad for us. So let's talk about bacteriophages for a bit. Bacteriophages are viruses that are going to infect bacteria. Notice they're called phages. Again, phage means to eat. These are not actually eating bacteria. They're actually just using the bacteria as a host. we'll show you an image here in a minute that will explain why we initially call them bacteriophages. But the idea is, again, bacteriophages are viruses that infect bacteria. So here's a great example. This is the T. even bacteriophage. Again, the T. evens typically infect E. coli cells. And naturally, E. coli is something that we've worked with for a long time in the scientific world and we know a lot about. So conveniently, that was the first phage that we started looking at, was the one that infected E. coli, the thing we knew a lot about. And so naturally now we know a lot more about the T. even phages than we do the others. So this is the bacteriophage life cycle. Notice it's very similar to what we've done before with the adsorption, penetration, uncoating, synthesis, assembly, and release, with some few minor differences. So let's start off with the traditional lytic cycle, which is that full replication cycle. So we can see here we're going to go around this pathway here, which is again the lytic cycle. And notice we do start with adsorption, and adsorption is again attachment. But one interesting thing here is penetration and uncoating happen at the same time here. So we just sum it up as penetration, okay? And so naturally the progression again continues, where we start to see the genetic materials copied and turned into parts. So we make our parts and then we start to assemble them. So assembly, and then we have now these mature viruses that are ready to leave and release. So we still have adsorption, penetration, uncoating, synthesis, assembly, and then release. One interesting thing to note here is many times these T. evens cause the lysis of the cell. So they cause the cells to explode. So we just discussed a minute ago the idea of the bacteriophage, or of a provirus, but there are some bacteriophages that are what we call provirus or prophages, that again will take, and instead of fully going through the lytic cycle initially, they will take a little detour here we call the lysogenic state. And the idea is the lysogenic state is whenever the virus, once it gets in, incorporates the DNA into the host DNA. And so again, it's just going to hang out until naturally the environment is now conducive to them coming out and producing more viruses. So again, the lysogenic state is a temporary stop-off point that then leads back into the lytic cycle at another point. So this is that image I was alluding to that was illustrating again the kind of process of why we thought the bacteriophages ate bacteria. And you can see there are tons of phages all over this thing. And notice there's a huge chunk of the bacteria missing. And it's now, hopefully you recognize, it's because the cells lyse whenever the new viruses spill out. So again, these viruses aren't eating up the cell, they're using them for reproduction, and then they're causing the cell to explode as the new viruses leave. So naturally, again, the lytic cycle is that traditional get in, copy your genetic material and your parts, assemble the parts, and then get out. The lysogenic cycle, or lysogenic state, on the other hand, is where again the genetic material is incorporated into the host DNA, which is kind of a state of dormancy. And again, when the environment becomes more conducive for them, again the genetic material will be taken out of the host and then turned into parts and pieces, and we go right back into the lytic phase or lytic cycle. So temperate phages are the next thing we should talk about. They have basically the ability to go through adsorption and penetration, but again go into that lysogenic state. And so the prophage was the one I was alluding to earlier. Again, a prophage will actually take its DNA and put it in the host DNA. So it'll split the DNA of the host and then put their DNA in there. And like I said before, the DNA is just going to sit there. The cell is technically infected, but again, we're not producing any new viruses right now because the DNA is just incorporated in the host DNA. Whenever we see that DNA reactivated to leading to more viral production is an act called induction. So induction is the activation of that DNA that leads now to, again, replication of more viruses. One of the problems that we see with this lysogenic idea is that sometimes we accidentally cut out more DNA, or really DNA that's not even viral DNA, and so we can end up transferring genes, resulting in something called lysogenic conversion. So this is new traits that will be actually passed from one host cell to another. And so there are several examples of this, like diphtheria toxin, cholera toxin, even botulism toxins. Again, were kind of spliced together, pulled from previous hosts. So again, here's our steps of, again, viral replication. This is, again, comparison between the bacteriophages, which are bacteria viruses, and then, again, the animal viruses that infect us. And so you can go through here and look at how they handle adsorption, how they can handle things like penetration, your synthesis, assembly, and release as well. So the idea here is you can look through this and try to understand that there are, again, many similarities, but some slight differences as well. So I'm not going to care a heck of a lot about this last bit. Again, cultivating viruses is important for research and really learning all the important stuff about viruses overall. But again, you guys aren't going to do research typically, and so the idea of going into excruciating detail of what this is, it doesn't make a lot of sense. But I do want to stress two words to you here, and that's in vivo and in vitro. Okay, I think the book does a pretty crappy job of this, so we're going to just simplify it down. Okay, many of you know what in vitro fertilization is, right? It's where we take the egg and the sperm out, and we actually conceive the child in the laboratory. So conception and in vitro actually happens in the lab. Well, in vitro literally means in glass or in the lab. And so the idea is the in vitro methods here is literally methods of growing viruses in the laboratory setting. In vivo, on the other hand, is more like the natural setting. So I'm not going to discuss with you how in vivo fertilization works. I think a few of you may know. But the idea here is that in vivo methods in viral research include actually using animal hosts to grow the virus. Again, different viruses, since they're very particular, very specific, will require different hosts or even different structures within the host. So it's important, again, to understand that viruses tend to be very specific. So if you have a virus that only affects, again, mammals, you're going to have to use a mammalian host. Or if you, again, have a virus that infects birds, you're going to have to use birds as hosts. Again, if it only affects the liver of an organism, then you're going to have to typically put the virus in the liver. So, one thing I do want to spend just a second on is this idea about using bird embryos. These are basically the equivalent of kind of like a giant cell. It's really not, but there's enough tissues here in rapid development that we can actually get a lot done in a small space. And eggs are nice and compact, again, with lots of resources. And so the idea is using eggs to grow viruses is actually a very good strategy. And so many viruses are grown this way. But why I bring this up and why it's really relevant to us is this idea that bird embryos or eggs are something that some people can be allergic to. And so the understanding that having the virus grown in an egg could elicit allergic response in a human host. And so in the cases of vaccines that are produced in eggs, well, if someone's allergic to eggs, they can't have that vaccine. But cell cultures or tissue cultures are a newer technique that is, again, gaining lots of popularity because now we can continually work with cell lines that are, in fact, many times human. And so the idea there is we can have a continual line that is going to not typically cause allergic response or issues. And so, again, cell cultures typically used in the place of many animals so that we can. So you can see here the basic comparison of normal cells versus infected cells. Notice how in this particular situation the cells are actually going to clump together. You can see nuclei are actually stained purple here, right? And so whenever you see this mass of nuclei, that's suggesting to you that the cells are kind of pulling all together. So primary cell cultures typically are freshly isolated from animals and placed in a growth medium. They'll go through mitotic divisions and will retain those characteristics of the normal cells, but they're a little more difficult to work with, again, as we're just pulling them from a host. The continuous cell cultures, on the other hand, are something we're much more interested in. With continuous cell cultures, again, we have this line that will grow continually forever as long as we feed them. And so what we get is a cell type that basically divides uncontrollably. And really, hopefully, by me making that statement, you think of cancer. And it's important to understand that most of these continuous cell cultures did initially come from or are still considered to be cancerous cells. Again, as long as you feed those cells the resources they need, they will continuously divide without any restrictions. So let's move on and talk about growing basically phages. Again, growing bacteria is pretty easy, so growing viruses that feed on bacteria is also pretty easy. But what happens again is there's typically damage done to the bacterial cells whenever the virus goes to leave. And that damage can result in these holes or patches that form in the sheet of bacterial growth, lawn growth, and we call those little patches plaques. And really what that amounts to is that's a whole lot of dead cells. And again, those cells are dead because the virus killed them in their replication. So that really ends, again, that culturing idea of viruses. Again, I don't care that much about that in terms of testing on that topic, but there are some other topics that we do need to finish up with here. And the first one I want to talk to you about is the really prions. Prions are going to be not cells, they're not viruses. What a prion actually is going to be a protein that causes basically misfolding and tissue damage of other proteins. And so the idea here is these prions are rogue proteins, and these rogue proteins are incredibly dangerous because they cause something called typically spongiform encephalopathies. And so hopefully, again, you can break that apart and understand we're talking about basically infections of the brain, but the word spongiform is suggesting that the brain takes a sponge-like appearance. And so what this really does is these misfolded proteins get into the brain and they cause basically the brain tissue to start to, develop holes in it is probably the best way to put it. So you get all these little pockets where the damage has been done. Naturally, creating holes in the brain is going to be incredibly bad for the host, and again, will most likely kill the host, but before the host dies, typically they go mad. And so by far, the most common example that people know about is mad cow disease. So mad cow disease is actually a prion disease. But there's a kind of a human equivalent here in Creutzfeldt-Jakob's disease, or CJD. Again, it does attack the central nervous system and the brain of a person and will lead to that degeneration and then ultimately death. We don't really fully know where it comes from, how it's completely transmitted, but we do understand that typically contact with a prion infected person or even their brain, specifically their brain, is a more likely means of transmission. And so the idea is typically as long as you don't eat people or their brains, you're probably usually pretty safe. But prions are so potentially deadly and so, again, at this point, understudied, we don't typically take risks. So whenever we see outbreaks of prion diseases, we have to immediately shut down the place and typically try to remove the prion from the environment. If you recall before, I said that endospores are the hardest living thing to remove. Well, just a single protein isn't considered to be alive. And so the idea here is prions are actually much more difficult to remove from the environment than even endospores. And so understand that this is no joke. Whenever we see a prion disease, we take it very serious, and we typically have to, again, go through extensive measures to remove it from the environment. So naturally, we still don't know a lot about these, as I've been alluding to. Again, they're protein made. And so the idea is that, again, these proteins somehow misfolded at some given point. But again, we don't know the mechanisms of how they hijack and produce more misfolded proteins. So again, these are still very new topics to us that we're trying to really understand. Viroids is really one of the last things we're going to talk about here. With a viroid, viruses are always going to be an important topic because viruses do infect lots and lots of people. We don't actually know how many people because information in terms of infections doesn't always make it to us. And most people don't go to the hospital for things like a cold, right? And so we don't know exactly how many people get sick, but we do estimate it's significant amounts every year. There are things that are found basically everywhere people are, colds, the flu, herpes. Anywhere someone's putting something in something else, herpes shows up. And so the idea here is, again, we don't know accurate numbers of a lot of these things, but from our estimates, what we can observe in the areas that do report, the incidents are pretty high. There are some things, though, that are regionally locked, and we'll talk a little more about them later, things like yellow fever and dengue. These are things that, unfortunately, are particularly not good, in that they are very, very, very bad for the health of the person infected with them. And unfortunately, these are places that often have many more difficulties in terms of getting people the treatment they need. So I do want to point out there are some things that will typically have what we consider very high mortality rates. Rabies, for example. If you show signs and symptoms for rabies, there's literally nothing we can do for you. You will die. And so the idea is, if you are bitten by something that is rabid, please seek medical attention as quickly as you can. Again, if we get you the vaccine before the virus grows out of control, we can save your life. But if you wait and you start showing signs and symptoms, there is nothing we can do for you. There are other things that are still quite the mystery, things like Ebola. Ebola, for a very long time, we didn't know much about, because by the time we heard about it, it killed everybody. And so we'd go do postmortems, but we couldn't actually track actively transmission. We didn't understand, again, lots of mechanisms that allowed for it to stay in the population. And so the idea is, some things are more difficult because they're more likely to kill somebody. There are other things that can lead to, again, that long-term cell damage that leads to then potentially long-term disability. So polio was a good example. Since polio lyses nervous tissue, it would naturally destroy the ability to use nervous tissue for its function, which is typically things like movement. And so you had things like individuals that were paralyzed for life. And so the idea here is that viruses are a big topic because they can kill you, or even potentially create long-term life, disabilities that unfortunately there's nothing we can do for. And so again, take viral infections overall quite seriously. Do everything you can to avoid picking up infections like these. So one of the bigger problems that we should probably point out before we move to the end here is that viruses are going to change very rapidly. Since they don't follow the rules of life, they can change pretty uniquely, very quickly. And that really results in a lot of problems in trying to design therapies and vaccines for these things. The idea that, again, at any given point, the virus can become something very different, making the drugs not effective, is very relevant to medicine. And so, again, hopefully that helps you understand why we have annual flu vaccines, because the flu that affected us last year is most likely not the flu that we'll see this year, because over the course of the year it's changed so much genetically. But there is one interesting new technique that we're looking into called interferon therapies, or again, IFNs. These interferons are actually naturally produced by your cell and are mechanisms essentially to prevent viruses from taking hold and causing infection. And so we can artificially insert interferons into a person to prevent their cells from getting infected. And so the idea is potentially this is a topic that we could use to prevent infection.